Hi guys, welcome to the DNA Daily. Today we're looking at DNA replication and the classic experiment that led to the discovery of how DNA replicated. Don't forget that DNA replication happens for us during interphase when we are doubling up on the DNA in order to make more cells that are exactly the same. So, first step that was done in the experiment was to essentially get a source of DNA, which was from bacteria. Now, Michelson and Stahl, they grew bacteria in a nutrient broth that contained heavy nitrogen. So they used this heavy isotope of nitrogen, which was essentially going to be used by the bacteria to replicate itself. Now, I'll stop you here. We have essentially a double helix that is made of heavy nitrogen, and I'd like us to just go to the centrifuge, and what we're going to do, guys, is we're going to spin it. And what we do is when we spin it, we separate out things due to how heavy they are. So the guys span the nitrogen and span the DNA, and it all sunk to the bottom in their little centrifuge like that. Okay. The next step in the experiment was to then move this DNA, or move this bacteria rather, into a source of light nitrogen. So we can move that across into the light nitrogen. And essentially they allowed for one replication, and then they did a spin on the centrifuge. What happened was they got a line there, so it moved up from where it was originally. What does this tell us? It tells us this. It tells us that our DNA essentially split like that, and because it was grown in light nitrogen, it used light nitrogenous bases and light nucleotides to make its other strands like that. So you have a uniform mixture of DNA that's lighter than it was originally. Floats up a bit. Now we can go to the third stage. The scientists allowed this to replicate again. So we can take the strands like so from here before, and we allow them to replicate again, still in light nitrogen. This is what happens. DNA unzips, all the light nucleotides come in, and if I can get this right, bind to their complementary bases like so. Okay, not having a good time here, am I? Let's do that there. And there we go. Oh, we got one more to do. This is what happens. How do we know this is what happened? Well, they put it in the centrifuge again, and they got a line in there that was a bit lighter than the original heavy stuff, but the same as what was on here. But they also got a line that was a lot lighter. Okay, which showed us that every time DNA replicates, it keeps half of the original strand, and it builds off of that to make a new double helix. I hope that's clear enough. We can go through the details in a quiz.